Photosynthesis at its core is all about transforming solar energy, which is scattered, difficult to store, difficult to transport, into a storable, controllable, transportable form of energy in the form of chemical energy. And it's very interesting to understand what is the energy efficiency of this transformation process. How much energy goes in, how much energy comes out. What are plants operating at in terms of the efficiency of this transformation? That's what I want to go into in this video. I'm going to start off by looking at the stoichiometry of the processes of photosynthesis, both light and dark reactions. And then looking at that stoichiometry, we'll be able to break it down into an energy efficiency. So at the end of the day, we'll look at how much energy was in those photons, how much energy is in the glucose, and we'll start with some assumptions, and then we'll kind of tease out some more detailed elements that are often overlooked. So if we look at a plant leaf, usually we think about there being some top layer and some bottom layer cells. The top layers have more chloroplasts, and within those chloroplasts is these beautiful green little structures called thylakoids. Those thylakoids have a inside called the lumen and an outside called the stroma. And on the thylakoid surfaces will be the photosynthetic apparatus. So looking at that photosynthetic apparatus here, we can see a few main components and a few main processes. So I'll walk you through here. So this is representing a thylakoid. The inside is the lumen and the outside is the stroma. And we see a few main components: photosystem 2, cytochrome B6F, photosystem 1, and ATP synthase. The whole point of these reactions is basically to take solar energy, represented by these photons here, and pump in protons into the thylakoid, which are then only going to have one way to get out, through ATP synthase. So the protons are going to escape the positively charged, increasingly positively charged thylakoid lumen space to go into the stroma, and in so doing, they're going to rotate ATP synthase and produce ATP. And we'll explain the ratios of these processes in just a moment. And then another important part will be the production of the reducing factor NADPH. And NADPH and ATP will then go and contribute to the dark reactions, the Calvin cycle. So now looking over here at these different steps, we'll start off with water. So for every one water molecule that goes through this reaction, we're going to oxidize that water, and we're going to liberate 2 electrons that are going to be transferred to photosystem 2 via this triangle here, which represents the oxygen evolving complex. And those 2 electrons are going to be excited in photosystem 2 by some photons, and those electrons are then going to reduce something called plastoquinone, which is represented by this little oval. And plastoquinone is going to grab some protons from the stroma, from this outside space, and then it's going to drop those protons off into the lumen while transferring the electrons onto the continuation of this electron transport chain. Now, I just said 2 protons, but there's also something called a Q cycle taking place in B6F, which kind of amplifies the efficiency of this proton pumping, so that for every 2 electrons that go through this step, there's actually going to be an additional 1 proton that's going to be pumped in. So that means we get, for every 2 electrons, 2 protons for the plastoquinols, plus 1 proton for the Q cycle and B6F, giving us 3 protons. Now, as you can see, we also got 2 protons from the oxidation of water, because we have these 2 H pluses that were sitting here. And so we've accumulated 5 protons that are available for the so-called proton motive force. And you can imagine, as you keep running this, the inside becomes increasingly positively charged, and these things want to get out. So they're going to be getting out through ATP synthase. But let's, for now, continue on the journey of the electrons. So the electrons have now given up a fair bit of their energy to accomplish this, pumping of protons against a gradient, and they are now at a lower energy state, and they're going to need to get excited once more by photons, and that's going to take place in photosystem 1. So they get re-excited by 2 more photons, and those 2 electrons are now going to reduce through a ferredoxin and ferredoxin NADPH reductase. They're going to reduce NADP plus to NADPH, and there we go. Now we've got our NADPH and our ATP. Now we want to know how much of each of these do we have. So there's a little bit of variation in different organisms, and there's also, I think, different um, physiological conditions that would change these ratios. But a good rule of thumb is that the ATP synthase requires approximately 4 protons to be pumped through the ATP synthase to generate 1 ATP. So it's something like 12 to 14 protons per 3 ATP, and therefore 4 protons per ATP. So if we have 5 protons here going into the system where 4 are needed for ATP, we can say that we are getting 1.2 ATP for each water molecule that's oxidized. Good, so doing a quick recap now. We said that we put in 1 water molecule and we got out half of an oxygen molecule, and we liberated 2 electrons. Those 2 electrons went through the electron transfer chain and ended up producing 1 NADPH. This process pumped in a total of 5 protons, which allowed us to produce 1.2 ATP. And we had to excite each electron twice, right? We had to excite an electron at photosystem 2, and then we had to excite it again at photosystem 1. So in total, for 2E minus, we need 4 photons. And by the way, why do they call it photosystem 2 and photosystem 1 in this order? I think it's because photosystem 1 was actually discovered first and photosystem 2 was discovered second. So even though it comes before, the naming is unexpected. So, all right, we're now in the Calvin cycle. So let's think about what do we actually need? What ratio of these elements do we need? So the Calvin cycle is taking place in the stroma of the chloroplast, just outside of this thylakoid. And the input is going to be CO2, and the output is going to be 3-phosphoglyceraldehyde, and then eventually the production of glucose. So what do we need? Well, there's a few different steps. I don't want to go into the details of the Calvin cycle in this video, but this diagram here shows the reaction balanced for 3 CO2 going in. And you can see that we need ATP twice. We need ATP at this reduction stage, and then we need ATP again also in the regeneration stage, and we need NADPH only once in the reduction stage. So for 3 CO2, we needed 9 ATP and 6 NADPH. So if I was to multiply this by 2, because I want to balance things for 6 CO2, that allows me to solve it for 1 glucose, and then glucose is going to be an easy molecule to think about from an energy perspective. So if I need that 6 CO2, then just multiply these numbers by 2, right? So I'll need 12 ATP here, 12 NADPH here, and then another 6 ATP here. So in total, 12 plus 6, I need 18 ATP and 12 NADPH. Okay, great. So 12 NADPH, 18 ATP, and now we run into our little problem. And this would be 2. The problem is that we said that we were only producing 1.2 ATP and 1 NADPH. So we can see that the ratio that we need is actually 1.5 to 1, right? We need 1.5 times more ATP than we need of NADPH, but our light reactions are only producing 1.2 times that. So how do the thylakoids deal with this mismatch? On the one hand, the light reactions seem to be producing 1.2 ATP per NADPH. On the other hand, the dark reactions require 1.5 ATP per NADPH produced. So the way that the plants are going to do this is actually by something called cyclic electron flow. And cyclic electron flow basically moves electrons from photosystem 1 back into the plastoquinol pool that we talked about, and causes proton pumping without causing the reduction of NADP+. So it's going to amplify the production of the proton motive force that's going to generate more ATP without producing NADPH. That's how the cells are going to add a little bit more ATP and balance the stoichiometry to meet the requirements of the Calvin cycle. However, for the purpose of solving the energy efficiency, it's going to add a fair bit of complexity, whereas we could make an assumption right now. We could solve the whole energy efficiency question with an assumption, and then we could always go back later and add this cyclic electron flow aspect, and it will increase the accuracy. But we can really go through the whole methodology without having added this for now. And that's what I want to do here. So we're going to omit cyclic electron flow from this analysis. And what we're going to do is we're just going to make an assumption here. We're going to say, okay, instead, we know that it's 5 protons per water molecule, but let's just assume that it's 6 for the purpose of this first calculation. So this is not going to be correct, but it's going to be close enough. And that's going to allow us to do the calculation without getting entangled in too many complexities. If we were to make this assumption, so let me highlight it in purple. If we were to make this assumption, then we could actually now change this number, right? And we say, okay, actually this will be 1.5, right? Because we now have 6 protons, and it's 4 protons per ATP. So 6 protons going through, we're going to get 1.5 ATP. And now we have the right ratio, and in order to reach this 18 number and 12 number, well, we just need to run the cycle 12 times. In terms of the energy containing inputs, it's really just the photons. So we have to figure out how many photons we're putting in and what is the energy content per photon. And then we're going to look at the energy content of that glucose. And that's going to be our first line approximation of this. And then there's other things that we're going to add, for example, photorespiration and this cyclic electron flow issue that we mentioned. But for now, let's go back to these photons on the input side. So we need 12 times 4 photons to get to the requirements for our Calvin cycle. And so that's going to be 48 photons. And what is the energy content of each of these photons? Well, we are in photosynthetically active radiation range. That's 400 to 700 nanometer wavelength photons. And if we consider the average of that range, it's 550 nanometers. So the typical photon, we could say, is a 550 nanometer photon in this range. And using that wavelength, we can calculate the energy efficiency. 550 nanometers, a bit of an awkward number. So, within 10% accuracy, I think we could say that the typical photon is 500 nanometers. Okay. Now we need to use the equation for the energy content per photon, which is given as energy per photon is equal to Planck's constant, often written as h, times the frequency of that photon. So the higher the frequency, the higher the energy. And the frequency can also be written as the speed of light divided by the wavelength of the photon. So writing this, so the energy is, the Planck's constant is 6.6 times 10 to the negative 34. We'll just abbreviate or shorten that to 6.6 times 10 to the negative 34 joules second. And the speed of light can be written as 300 million meters per second. So million meters per second, 10 to the 6 meters per second. And then the speed of light, the wavelength is 500 nanometers. Now, we don't have comparable units. I'm going to start off by working on the frequency side. We don't have comparable units. We have meters on the top, nanometers on the bottom. I think it would be easiest to convert meters into nanometers, so I will do that. And we can just basically play with this number here. So how many nanometers are there per meter? Well, there's 1,000 millimeters per meter. There's 1,000 micrometers per millimeter, and then there's 1,000 nanometers per micrometer. So we have 1,000 times 1,000 times 1,000. That's 1 billion. So that's another 9 zeros to add, right? And now we are in nanometers. So let me just check that again, 300 million meters per second, and if we were to convert into nanometers, we would have to add another 9 zeros. Yeah, so that should be good. And so this is 15, and right away we also see that we can remove a few zeros here. So I'm going to remove these 2 zeros, and now we've got a little bit of a simplified equation here. So going to the next step, again, let's work on the frequency side first. So energy equals, I'll just leave the Planck's constant blank for now. So we have 3 over 5, that's 0.6, times 10 to the 15, but we can actually just make that 6 times 10 to the 14. And we cancel out the nanometers, we're left in per second, so that's a frequency now. And so we've got 6.6 times 10 to the negative 34 here, so we have 6.6, and the 10 to the negative 34, well, I can just add that here, right? So I just do minus 34. This is joule seconds. We've got seconds on the denominator, so seconds cancel out, and we're left with joules. So that's the remaining equation. So now, let's pull up the calculator. So 6.6 times 6, we'll call that 40, and then we've got 14 minus 34, which is minus 20, so we've got 40 times 10 to the minus 20 joules per photon, right? This is per 500 nanometer photon. Okay, so now we know the energy per photon, and we need to figure out how much energy is there in those 48 photons. So let's do energy total is equal to this number times 48. So 48 times this number. 48 photons times this number. Photons cancel out, and we are left with this. So let's do 48 times 40, 1920, we'll call that 1.9, times 10 to the negative 20, it's 1,920 times 10 to the negative 20 joules. Okay, and we can simplify that a little bit more. So first of all, we can round this to 1,900. And what if I were to remove some numbers here? So if I make this 1.9, then I'm dividing by 1,000. So if I'm dividing by 1,000, I need to multiply this side by 1,000, which would be the equivalent of turning this into negative 17 joules. Okay, so the total energy that we're putting in with these 48 photons is 1.9 times 10 to the negative 17 joules. Sounds like a pretty small number. And let's go look at how much energy is in our glucose. So at this stage, we could just look up on Google or GPT what is the energy content of 1 molecule of glucose. However, I like to have some rules of thumbs available to me, actually because we're relying so much on these kinds of technologies now, like GPT, that it's very helpful to have an intuition for some of these numbers and be able to detect when things are off. I think it's very dangerous to be too reliant on the technology and not have some kind of orders of magnitudes in our own mind. We're actually be able to kind of calculate things a little bit ourselves. So some of those baseline numbers that are useful is the energy content of carbohydrates and hydrocarbons. So carbohydrates, sugars, are typically 4 kcal per gram, right? And hydrocarbons or fats are 9 kcal per gram. And there's 4.184 kilojoules per kcal. So this is basically 16.7 kilojoules per gram of glucose. All right, and now we can also calculate the molar mass of glucose. We could just look it up, but it's very easy to calculate. So we know the equation of glucose is C6H12O6, and so the molar mass should be 6 times 12, there's 12 grams per mole for carbon, of course, plus 12 for those H's, and oxygen is 16 grams per mole, and we've got 6 of those. Oops, so we've got 60 plus 2 times 6, 72, plus 12, 84, and 96, 84 plus 96. That's basically 100 plus 84, so 184 minus 4 equals 180 grams per mole. Okay, pretty easy to calculate that. If you know a little bit of the molar masses by heart, which you kind of end up knowing if you're doing a fair bit of biology and chemical calculations. Now we need to calculate the energy per mole, right? Energy per mole. So we know that we have 16.7 kilojoules per gram. We want that kilojoules on the numerator, so we put that first. And we want mole on the numerator, on the denominator, so we're going to put that here. And that should give us, the grams should cancel out, so that'll give us kilojoules per mole of glucose. Let's pull up the calculator. So 16.7 times 180, 3,000. So that's 3,000 kilojoules per mole of glucose, and we can make that 3 times 10 to the 3 because that will probably be useful in the next step. Now this is pretty close to the sort of calorific calculated or experimental glucose energy content, which is around 2,890 or something like that. Kilojoules per mole. So we can work with this number. And now we need to go from here to the energy content per molecule of glucose. So there we're going to use Avogadro's number. So that's 6.022 times 10 to the 23, but we'll just call it 6 times 10 to the 23. That's molecules per mole, Avogadro's number. Where's my cursor? Avogadro. And from here, we want to get kilojoules per molecule. So I'm going to put this at the front. I'll have kilojoules on the numerator that way. And I need to actually flip this over, or I could just divide, but I like to keep staying in multiplication. That just makes it easier to avoid mistakes. There's a mistake. Touch molecules. All right, so now getting close to the end here, we need to calculate the value of this number. So be careful with the units. We've got 3 on the numerator there, and then we've got 6 on the denominator. So that's going to be 0.5 times 10 to the 3 minus 23. Units are kilojoules per molecule. Let's take this number and whittle this down a little bit. So what can we do with that number? Well, this is going to be minus 20. Okay. And this 0.5, I'd like to turn this into a 5. So I'm going to multiply this side by 10, and therefore I need to divide this side by 10 to keep it equal. And I'd also like to get rid of this kilojoules, so let's just write it out one more time. So I need to multiply by 1,000 to drop this kilojoule unit and keep it in joules. So multiplying by 1,000, we'll be turning this into minus 18. That's it. Now we need to calculate the energy efficiency. That's going to be the output, which is our glucose molecule, divided by the input, which was those 48 photons. So we've got our output here, 5 times 10 to the negative 18 joules, and we've got our input right here. Okay, so units cancel out, that's the first thing. And let's see what else we can do here. So units cancel out, and let's do, we've got basically 5 divided by 1.9. times 10 to the 18 minus 17 equals 5 divided by 1.9. 5 divided by 1.9 is 2.6. 2.6 times 10 to the, sorry, this is supposed to be minus 18. Minus 18 plus 17 is going to be minus 1, 10 to the negative 1, which is the same as 0.26. This is a unitless ratio, and so the energy efficiency is equal to 26%. So now we've calculated that 26%, that's the maximum photochemical energy efficiency of photosynthesis. But of course, we've made some simplifying assumptions. We've left a few things out, and those will be the subject of a subsequent video. But just to touch on them, to complete the story a little bit, well, one of those factors is the photosynthetically active radiation is only a fraction of the light that's actually hitting the photosystems, right? So we said that these photons are between 400 and 700 nanometers, and that's fine, but for every photon that's within that range that hits these systems, there's also going to be one photon that's outside that range, because the total range is much broader, and approximately 50% of the light that hits the terrestrial surface is going to be between 400 and 700 nanometers. So all the other photons that hit these photosystems are actually not going to contribute to glucose production. In fact, they could be counterproductive because they create heat. They can contribute to oxidative stress and so forth. But that's going to be one factor that, in practice actually reflects a lower energy efficiency compared to the calculation we've just done here. Another factor is this cyclical electron flow. We made this assumption, but in reality, we know that there must be some electrons that are cycling back around PS1 back into the plastoquinol pool, and that's going to require more photons to hit PS1. Interestingly enough, the plants have significantly more PS1 than they have PS2 because of this cyclic electron flow requirement, because they need to boost ATP production to meet the requirements of the Calvin cycle to get this to this 1.5 number that we mentioned. Another simplifying assumption here that we've made so far is we've omitted photorespiration. Photorespiration is a major contributor to inefficiency of plants. It's also a major avenue of research in a lot of different labs. And what happens in photorespiration, in a nutshell, is that the enzyme rubisco has carboxylase oxygenase activity. It will occasionally, instead of fixing CO2 as it's supposed to, it will fix O2, which is a similarly sized molecule and fits well into the reactive pocket. And when it does that, it actually ends up producing a toxic molecule called phosphoglycolate, which it needs to get rid of. And it's going to cost the ATP, and it's going to lose CO2 in the process of recovering the carbon in that phosphoglycolate and getting rid of the phosphoglycolate. So that's another inefficiency, and it becomes even more pronounced in conditions of low CO2, high oxygen environments. And so that's another thing that we can consider in the calculations. But what we've calculated here today is we've shown that with PAR photons, photosynthetically active photons, there will require basically 48 photons, at least 48 photons, to make one glucose molecule, and that would result in an energy efficiency of 26%. So that's an interesting number, because we can now compare that, for example, to photovoltaics, which have this Shockley-Queisser limit of 33% for single junction photovoltaics, and multi-junction photovoltaics can go much higher. But this gives us a sense of the difference the different maximum energy efficiencies of these two systems. An important caveat there is whenever people are comparing these energy efficiencies, we have to be very careful to remember that the plants are producing chemical energy, glucose, they could be producing hydrocarbons or proteins, things like this, whereas the photovoltaics are producing electricity. So in some cases, one might be a much more valuable form of energy than the others. And just be careful, we're not exactly comparing apples to apples when we compare photovoltaics and plants directly head on like that. We have to think about the different form of energy.